AP 120, Chapter 15, Topics, Small Intestine and Cholesterol. So the small intestine is the largest region of the alimentary canal and is about 20 feet long. It has many functions. It receives secretions from the pancreas and liver, pancreatic juice and bile. It completes the digestion of the nutrients, absorbs the nutrients, and then transports the remaining residues into the large intestine. The smallest portion of the small intestine is the duodenum, comes directly after the stomach and forms a short C-shaped curve or turn. The duodenum receives the chyme from the stomach, the pancreatic juice, and the bile. Then in the upper left quadrant, we can find the jejunum. The jejunum comes directly after the duodenum and is relatively mobile. It wiggles around, as it were. And then the remaining portion of the small intestine is the ileum. Ileum comes directly after the jejunum, found mostly in the lower right quadrant, is mobile, is the longest portion, and ends in a structure called the ileocecal sphincter. The ileocecal sphincter controls the release of materials into the large intestine. So here is the small intestine, and as we can see, it's bound up and suspended from the posterior abdominal wall by part of the peritoneum, specifically the part called the mesentery. And we can see the mesentery is extremely important because not only does it surround the small intestine and attach it to the posterior abdominal wall, but it is also what is bringing various blood vessels, nerves, and lymphatic vessels to and from the small intestine. All right, here is the wall of the small intestine. The innermost layer is, of course, the mucosa next to the lumen or space inside of the small intestine. The mucosa has these finger-like projections that push into that space called the villi, many villi, one villus, and they increase the surface area of the lining of the small intestine, which then increases absorption. And also the villi also aid in mixing actions such as segmentation. Here is one villus. When we look at it, we can see that it is lined with simple columnar epithelium, and also associated with the simple columnar epithelium are the goblet cells that produce mucus. It has many, many blood capillaries on the inside of the villus, as well as one lymphatic structure called the lacteal. And the lacteal is what carries absorbed lipids, the fats. So the lymphatic system carries the fats from the small intestine, while the capillaries, the blood capillaries of the cardiovascular system, carries the rest of the nutrients. Again, there are many goblet cells that produce mucus to provide lubrication. And at the very base of the villi, we find a small invagination referred to as the intestinal gland. The intestinal gland secretes a large amount of watery fluid to keep the contents of the small intestine liquidy. All right, when we look at individual cells of the small intestine, we see that those simple columnar epithelial cells have these little apical projections called microvilli. The microvilli, again, are increasing surface area, which then increases absorption of nutrients. In addition, there are enzymes embedded in the macrophyllae that finish the digestion process. So peptidases that break down dipeptides into single amino acids, various enzymes, sucrase, maltase, lactase that break down disaccharides into monosaccharides, some intestinal lipases that help to further break down triglycerides into fatty acids and glycerol. And this allows for the complete chemical digestion of nutrients, allowing them now to be absorbed. So the monosaccharides The simple sugars and the amino acids and nucleotides, they all go straight into the blood capillaries. So they travel via active transport and or facilitated diffusion into and through the epithelial lining and then enter the bloodstream. Water also is absorbed by the lining of the small intestine, absorbs by osmosis, and that goes into the bloodstream. And electrolytes are also absorbed, so salts are also absorbed by active transport and sent to the bloodstream. For the fatty acids, though, things are a little more complicated because, again, fatty acids are hydrophobic. So first, the fatty acids diffuse into the cell. They're able to cross the cell membrane because they are hydrophobic. Then they end up in the smooth endoplasmic reticulum, where they are used to form new triglycerides that the body desires. The triglycerides, the fats, are then encased in proteins, forming a structure called a chylomicron. And it is these chylomicrons, these fats surrounded by proteins, that are then sent to the lacteals, sent into the lymphatic system. And then the lymph carrying the chylomicrons will eventually make it to the bloodstream and return these chylomicrons to the bloodstream directly. So it takes a while, but the chylomicrons will end up eventually in the bloodstream. All right, chylomicrons, important for transporting dietary fats to the muscles and the adipose tissue. And this, of course, includes cholesterol that we eat. Cholesterol we eat is a lipid, so it's also going to travel via these protein-fat complexes. And again, why do we need alternate ways of transporting fats? It is because fats are insoluble in water. They are hydrophobic, so they have trouble traveling in the watery plasma or lymph fluid. Instead, they have to form these weird protein fat complexes. All right, the liver is also involved in transporting fats. This is because the liver, as we know, is involved in both the storage of some fatty materials and in various metabolic functions that include converting things into fats or converting fats into glucose. So the liver is involved in the use of fats. So one important thing the liver does is transporting, sending out triglycerides that it has produced. So the liver produces these triglycerides, wants to send them out in the bloodstream. It sends them out with cholesterol, which we know is another lipid. So cholesterol and triglycerides are not traveling, and they are surrounded by proteins. And they form what are called VLDLs, very low density lipoproteins. So the lipids are surrounded by proteins so they can travel in the bloodstream. The VLDLs reach the various tissues that want the triglycerides, and the triglycerides are then deposited in, say, the fat cells, the adipose cells, or in the muscle tissue. However, the story's not done. The VLDLs then become LDLs, low-density lipoproteins. Loss of triglycerides, triglycerides are gone, but there is still cholesterol surrounded by proteins. And the cholesterol is continued to be delivered to various tissues of the body. So the LDLs continue traveling, continue delivering cholesterol to various tissues of the body. And this is why LDLs are referred to as the bad kind of cholesterol, because it is the structures that are delivering cholesterol to the tissues of the body. However, we do need a certain amount of cholesterol in our tissues, so it's not truly bad. All right, so cells remove LDL from the blood by receptor-mediated endocytosis. Cholesterol is important because our cells need it in our cell membranes to give the cell membranes proper rigidity. Although, of course, if you get too much cholesterol, then it can lead to problems. Cell membranes becoming too rigid, becoming more fragile because of the rigidity, and potentially leading to the formation of fatty plaques inside of arteries. So again, LDLs are being taken in by cells so that the cells can take that cholesterol. And finally, after all that, we now have the high density lipoproteins, high density HDL lipoproteins. Its function is to remove cholesterol from tissues and take it to the liver. So the liver produces HDL proteins. They go out to the tissues and collect cholesterol and bring cholesterol back to the liver. So this is referred to as the good cholesterol because it is the removal of cholesterol from the tissues and taking it back to the liver. And then the liver will secrete the excess cholesterol in the form of bile. That is the complicated route that cholesterol gets to the tissues and then gets back to the liver. So here we have lipids coming from the small intestines, lipids in the form of chylomicrons, proteins surrounding the lipids. These are the dietary lipids, and the cholesterol we consumed goes eventually through the lymphatic system into the bloodstream, travels to the various tissues, including muscle tissue and adipose tissue, delivering the lipids and also the cholesterol, and then the proteins that are left over go back to the liver. Then we have the lipids and cholesterol produced by the liver. And the liver will then send out these VLDLs. This is the lipids and cholesterol. They go into the bloodstream, travel to the tissues. The VLDLs get rid of their triglycerides, get rid of their fats, their lipids, but leave the cholesterol behind, becoming LDLs. And then the LDLs will go into various tissues, delivering the cholesterol. So again, the LDLs deliver cholesterol to the tissues of the body, and then the remaining proteins go back to the liver. And then finally, we have the HDLs. The proteins for the HDLs are produced by the liver. They go out to the tissues. Then the HDLs will collect excess cholesterol from the tissues and transport excess cholesterol back to the liver. That is the very interesting process that lipids and cholesterol, which is technically a kind of lipid, are sent to the various tissues of the body, and in the case of cholesterol, also returned back to the liver to be excreted in bile. So then the bile sends the cholesterol into the small intestine. That then leads to the large intestine. Got to get rid of that excess cholesterol in bile. All right, movement of material in the small intestine. You got segmentation that helps to mix materials, and peristalsis that moves it along the length of the small intestine. This can take 3 to 10 hours for materials to move along the small intestine. Occasionally, there'll be a very strong peristaltic wave that will move things even faster. This can sometimes lead to diarrhea as too much fluidy mixture enters the large intestine. At the end of the small intestine is a structure called the Ileocecal Sphincter between the small intestine's ileum and the large intestine's cecum, Ileocecal Sphincter, and that controls the movement of materials from the small intestine into the large intestine. And this often occurs after eating. So the material, which we're going to now call feces, enters the large intestine, and this usually happens after you have eaten a big meal. And that is it for this part of the lecture.